Our next speaker is here in person. It's George Janssens from Amsterdam UMC.
All right, great. Thank you for the invitation and the opportunity to give this talk. I'd like to thank the organizers for that. So as you can maybe tell from the title, I've got a clock for everyone. I have a drug for everyone. So I hope you'll enjoy. Before I jump into how we got to this particular research question, I want to explain a bit of the background, also because I'm setting up my own independent research lab. It's a good way to introduce you a bit into what we're doing. So we work on three main things, shamelessly organized by publications. So molecular aging mechanisms, so that's we're working with mitochondrial biology, protein translation, also biomarkers of aging, which we are doing with metabolomics and lipidomics. And probably our main focus is on pharmaceutical interventions, which is mainly to find probes to interact with the aging process, but also hopefully to find low side effect drugs for people. Now, the work I'll tell you about today is a combination of two of these, the biomarkers and the pharmaceutical interventions. It's just been out in a wonderful journal called Frontiers in Aging, which I recommend to everyone. And you can also see a first author's name appearing a lot on this list. That's Rebecca, who's in the audience now. She's in her last year of PhD, also will be on the job market for a postdoc, so I recommend you to talk to her. But now let's get into this paper and this clock. So we have seen a lot of molecular aging clocks, and our goal was to do something that's as downstream in the aging process as possible, which for us meant movement. You can argue that your ability to move is actually the best marker for your health and how you're doing during aging. So we wanted to make a clock based on movement patterns with the hope that we would find individuals who were by our clock, biologically younger, and then we would look for factors associated with that biological young phenotype, either drugs or nutritional components. So we used a database called NHANES, which is quite famous and popular. There are thousands of participants per study round. A survey including many things. For our purposes, age, drugs taken, and foods eaten are useful. And there's also paired mortality data for the older years. And especially important, there are two consecutive years, 2003, 2005, which had accelerometer measures. So that's basically what's in your Fitbit or your phone or your smartwatch, tracking your movement patterns. I'll also mention that while we were working on this over a couple of years, at least 4 papers came out showing that you can also predict biological age from movement patterns. The top 3 are actually from the same database we used. So it's quite a robust phenotype, actually, and I recommend you to look at those papers too, which are very informative. Our goal was then to use that to find factors associated with it, which I'll walk you through now. So first, the data. Always visualize your data to see what's underlying things. So this is how the accelerometry data looks. 7 consecutive days, in their case, a device worn around the leg. And I have on the left average 18-year-old, on the right an average 80-year-old. You can already see differences in the patterns. So what we did is we took this minute-based accelerometry data, we compressed it with summarizing statistics to make it a bit easier to make models on. Summarizing it with variance, hourly variance, and hourly maximum intensity. And that gives us already a pretty good distinction between the ages. We took a pretty gold standard approach to building a model, which is you start with one of your data sets, you split it into training, 70%, and testing. You build your models. For us, we used random forests, so these are decision trees, many of them, that you grow over the data, and then we have a subset that we serve for validation. And then we bring in fully independent data set, 2005, which we use to make the prediction, and then we compare our predicted age versus our chronological age, which you can see here. So jumping straight into the results, the output. So we're looking now at the 2005 data, and this is our chronological versus our predicted age. One thing I'll mention is we do a normalization step on the data. So if you look very closely at a lot of the age predictors, you can actually see some skewing in the predictions. So you have, for example, systematic underrepresentation of biological age for younger people and overrepresentation for older people, or vice versa. So that's fine for different purposes, but for us, we wanted to look at things related to delta age. So you're subtracting predicted age versus actual age, and if you don't have a one-to-one relationship, you're actually looking at factors associated just with chronological age. So for us, we then normalized. We end up with our mean squared error of about 8 years, which is, of course, a bit more than the DNA methylation clocks, which have 2 to 4, as we've seen, but around the range of the blood biochemistry markers. And maybe what's funny to say somehow is in the field we got away with saying this error in our model, right? It's not our model that's wrong, but it's your body that's wrong. And we then convert this axis from predicted age into biological age, saying that it wasn't us in our computation, but it was your body. And that's a big leap of faith, but it's okay to do it if you can prove that people that you predicted to be biologically older might be more likely to be dead in a follow-up period, for example, because they are biologically older. So that's what we looked at. So we have our data here stratified across different decades of the participants, and we looked just after the follow-up period, which is about 10 years, who was found alive and who was found dead, and how do the delta ages look. So a positive delta age is biologically older. And we can see, well, starting around 40, we have a pretty consistent, significantly older, higher delta ages from our model. So for us, we say that's good. That's a validation that we can call it that there's something biological going on. And then we can look at what are the factors involved. So we looked at, well, we looked at nutritional components, which I won't go into. Maybe just mention our top hit was fiber. So that says something about also your microbiome influencing things. But I'll go straight to drugs. So if you look at the participants here, we have the different decades here and the number of drugs they take. So if you're around 70, you might be taking 5 different drugs. And we used then 70 plus as our age group we were looking at, also because our original goal was to look at drug synergies. So 2, 3, 4 drugs together, how do those relate to decelerated aging? This data set ended up not being statistically strong enough to make those claims, but we went on anyways to look at single drug interactions. We did that by looking at the distribution of the delta ages of the users of a drug compared to all the other users. So here you have Doxazosin, for example, which is actually the top hit, and you see that the users tend to have a negative delta age. So they are, according to our model, biologically younger and maybe have some preserved movement ability with age. So if we can rank all the drugs like that from the database, where there are hundreds, we get only Doxazosin actually passing a p-value of 0.01, which if this is, I'll mention, an uncorrected p-value, so if you correct for multiple hypothesis testing on the hundreds of drugs, everything falls out. But we were still interested in this because there was a paper from the Petroshek lab who showed in some years ago in a big screen of over 1,000 compounds, they identified about 50 that was extending lifespan in worms, and one of them was Doxazosin, which they didn't go into more. But we were interested because that might give us hope that it's also do something for healthspan, which is really what our readout is for. So we also went to worms, and we treated worms with Doxazosin, and we looked at their ability to move with age. Here you have on day 9, which you could say is rather old for a worm. The ones who were given Doxazosin had higher speeds of movement on an agar plate. So that's a health span measure showing it's preserving their health span with aging. We then collaborated with NemaLife, who they run microfluidics for worm lifespans. And they also confirmed the lifespan extension. And then you can also look in their setup at the percent of highly active animals, which starts at around 80% in the population. And Doxazosin, through this measure, also was preserving high motility of the animals through aging. So again, showing preserved health span. So at this point, you might ask yourself, what is Doxazosin? It is an alpha blocker that's used for prostatic hyperplasia and also for hypertension. And in a lot of studies, you find that it has protective effects on other organs, which you might suspect from anti-hypertension drugs. But that's not relevant for worms who don't have hypertension. One paper, though, was published this year, showed that users of doxazosin were protected against developing Parkinson's. So Parkinson's is the biggest risk factor is age. So that gave us insight, and they suggested it was the glycolysis enhancing effect. So that's the direction we're now looking at for this drug and trying to find the actual mechanism in worms. And with that, I'm just going to summarize what we've done. So we had a population-level data set, which we had different factors, food and drugs. We used a biological aging score related to movement to find things that were associated to better movement with aging. And we then went through and ranked everything using delta H to find factors associated to better health or aging. So that's the general approach that we're also now taking on a number of other cohorts that we're generating ourselves and bigger data sets. So with that, I'd like to thank, of course, Rebecca, I've mentioned, Rikeld Houtkoper and his team, who I'm embedded in their group. They give a lot of input on this, and the team at Nimalife, and then the funding bodies, which are at the bottom here. And thank you all.
Thanks for a very interesting talk. Do we have a question in the audience? Otherwise, we will take one from Joris Deelen from Slack. Is it possible to distinguish the type of exercise from the wearable devices to determine what type of exercise works best for predicting biological age?
So at this moment, all we can really narrow it down to is the variance of your movement. So if in 1 hour you're having a lot of diversity in your movement, that's the resolution we have to say that that's contributing to having a lower biological aging score.
Okay, thanks. And there are more questions on Slack. Maybe you can also just go online to reply to them. But thanks again.
Thank you.